Hi, and welcome to this presentation entitled Making Sense of the Science of WM. My name is Zachary Hunter, and I work at the Bing Center for Waldenstrom's Macroglobulinemia at the Dana-Farber Cancer Institute in Boston. And my job is really to coordinate most of the genomic efforts, epigenomic efforts, and big data science that happens at the center. Now, this talk is actually going to be broken up into three distinct sections. First, we're going to be talking about just basic biology and basic science concepts that will allow us to talk about the role of mutations in cancer. We'll then switch gears and talk about how these individual concepts play out in the case of Waldenstrom's macroglobulinemia. And finally, we're going to talk more about how they form signaling networks, why signaling networks are important, what we mean by that, and how they're used to develop new therapeutics in Waldenstrom's and for other cancers as well. And then finally, we'll talk about how all these findings together have really changed the landscape of clinical trials and therapeutics that are being developed for Waldenstrom's macroglobulinemia patients. The first section is understanding genetics. Now, if you haven't had biology classes for a while, don't worry about it. Genetics is easy, and I really mean that. This is not a complicated concept. There's just a lot of small parts, figuratively and literally. So, the whole genome is really just made up of 4 distinct nucleotides, all strung together into 23 long strings. And that's it. That's the whole thing. You get one copy of this from your mom, you get one copy of this from your dad, and that's your genome. Full stop. Now, the details, of course, like everything else, are always a little bit more interesting. So these nucleotides are really remarkable molecules. Not only are they distinct from each other, but they're actually designed to pair together. Specifically, the nucleotides, which are abbreviated A, G, C, and T, are designed in such a way that G's pair with C's and A's with T's. This means we can come up with 2 anti-parallel strands of DNA that are perfectly complementary to each other. Now, this is important because not only is it incredibly chemically stable, it means that if something happens and one side is damaged, we can use the other side as a template to repair it. This is one of the reasons why we can find, and diagnose, for instance, genetic diseases in mummies, or try to recreate dinosaurs or woolly mammoths from ancient DNA sources. And these are actually real things that people are trying to do. Now, DNA itself is just this string of code. But what makes the code interesting is that sections of it are really instructions, mostly instructions on how to build things, so particularly proteins. And these sections of instructions in the DNA, we just call genes. Everything that's not a gene is typically either structure for the DNA or really some regulatory information about when a gene should be used or not used. Now, you can imagine the DNA as being like a master blueprint for a building. Let's say we're building a skyscraper. Well, we don't want to take our master blueprint and be running up and down the elevators, going to every floor and to every work person on that floor, being like, hey, Joe, like, this is what you need to do today. No, right? You make a copy, right? Relevant copy that each individual person needs. You don't need the whole thing. And that's how the DNA works as well. In order to protect it and make sure it is available to the cell, little copies of it are made, particularly of the instructions, and these copies are called RNA. It's a slightly different molecule, but made up of basically the same nucleotides. The only difference here is that the T's are now called U's for uracil, but are otherwise pretty much identical, and they're single-stranded and a little bit less stable. So these instructions are then sent out in the cell to go do work. And that really is genomics in a nutshell. Now, okay, so I said there's 4 different bases. You can talk about 4 different letters. Turns out, though, that there are 3 billion of them in the human genome. When you have 3 billion simple things, it turns out you can do some pretty complicated biology with this. And as an example, I'd like to think about computers. Computers really have a similar system, right? They have 2 bases, 1s and 0s. And with those 2 bases, with enough of them, you can make some very complicated behaviors. And like here, we're looking actually at the IBM Watson program that was able to beat all the human competitors in Jeopardy. So pretty impressive stuff from just 2 bases. So imagine what you could do with 4. And this is really a property in biology is when you have a bunch of very simple things together, you get very complicated emergent behavior. But even so, try not to be overwhelmed and just focus on the fact that every individual piece is actually quite simple. So let's take the whole picture together at once. So there's DNA, it stores information, is used as a template for RNA. RNA is transcribed from these regions that encode for instructions called genes. And they're really instructions on how to make protein. And so the protein does work, provides structure, and really does everything that the cell needs to respond to its environment. Now, there's some interesting problems that arise from all this. 3 billion is a very big number. And again, you have 2 copies of it, 1 from each parent. Now, if we were to take all that out, that's 40 inches strung end to end. Now, even with very, very impressive data compression, that's pretty hard to fit into every single cell in your body, let alone into a small compartment inside that cell, which is actually where the DNA resides. It's sort of an area called the nucleus of the cell. Now, this is a very tightly controlled process, and this actually leads to some very interesting consequences. In fact, the DNA is wound up so tightly that it basically can't be used, and it's invisible to most of the cells. Only parts of it are unwound, which means that when you think about why is it that like your skin, your eyes, your liver, and your heart all may respond differently to the exact same stimulus, even though they have identical DNA, they actually have different sections of the DNA unwound at any given time, which means that their effective genome, the genes that they're allowed to see and use, are effectively quite different. Now, to further help this sort of specialization in programming, there's actually chemical annotations in the DNA that can be added or removed called methylation events. And these methylation events are really just chemical annotations on the DNA that prevent protein from interacting with it efficiently. Again, either hiding or highlighting the DNA from certain types of interactions, making genes available or unavailable based on what's needed by the cell at that moment. So if we take this all together, we can now sort of see that this is actually a dynamic process. You have DNA, which are really encoding for all the different tools that are possible. Parts of it are read off and called RNA. That makes protein. Protein infects epigenomics, so it controls which parts of the DNA are available and also which genes that need to be used at a given time. And it comes with a whole sort of cycle, an interactive cycle that we'll talk about here in just a minute. Now, thanks to next generation sequencing and improvements in computing and technology, we can actually measure these all at once. Now, these are called collectively as omes. So we have the genome, which is the study of all the DNA in an individual person. And we can do like whole genome sequencing now of a person or of a tumor. We have the transcriptome, which is all the RNA that's being used in the cell at an individual like snapshot in a point of time. It's going to be very, very useful to understand how genes are being used by a healthy donor cell or by a WM cell. And finally, we have the proteome, which is sort of all the proteins, where they are located in the cell, how they are being used, and what they're doing. And last but not least, we have the epigenome, which is which sections of the DNA have been unwound and are available to the cell, and which ones have been chemically annotated in such a way that they're no longer available, even though they are open. Now, this is a lot to take in. But one of the things that people often get confused by is that if the DNA is sort of the master template for everything, why doesn't it just not control every aspect of who you are, who your friends are, what your favorite ice cream is? Well, really think of DNA as more like a computer operating system. For instance, everyone who has like a laptop with a Windows installed, for instance, really has identical, their laptop has identical DNA. They're all clones of each other, right? But none of them are used in the same way because they're all interacting with their environment differently. They're experiencing different environments because each one of you are different. Some people are playing games, or they're doing email, chatting, TikTok, who knows? But that is sort of the point. And as a matter of fact, when you look at identical twins, they start off being incredibly identical and very difficult to distinguish from each other. And this is because the DNA and the tools that they have are in fact the same. But as they start interacting with their environment and make different choices about how to use those tools, they will actually start to slowly differentiate from each other. And by the end of their lives, identical twins are often quite different from each other and are very easy to tell apart. And this is really the most critical part of biology, is understanding this cycle of protein to DNA to RNA to protein and back again, as this constant dance as the cells, the organs, and you yourself respond to the environment. Now, I've talked a bit about this idea that DNA encodes for RNA, which encodes for protein, but that's kind of hand-wavy, almost magical, right? So somehow you go from this code of 4 different bases to constructing these proteins. But turns out it's actually fairly straightforward. There's a transcriptional start site and a sort of place in the transcript that indicates where it is that the transcript should start to be read. After that, it's actually read in groups of 3 bases called codons. And we can actually just do this right now. Let's try a few. Here's a little codon chart that can help us understand how to translate from RNA into proteins. And proteins are actually just chains of amino acids. You may have heard of amino acids, health advertising and stuff like that. They're just the building blocks of proteins, again, that are coded for by the genes in your DNA. So let's take a codon. In this wheel, we start in the center and we work our way out. So let's say we had a codon that read C, U, which would be T in the DNA, or pretty much anything. Well, that's going to give us leucine. Now imagine that there was an event. Let's say, who knows, like a little bolt of UV light comes down, hits the DNA, and what was a T is now a C. What would happen? What would the impact of that be? We can look. So we have C, C, and then again, pretty much anything else, and we're going to get proline. So by changing just one base in the gene, we were able to change the coding for this one amino acid from leucine to proline. And that, in fact, can have a lot of consequences for the structure of the gene. So those simple substitution events, also called single nucleotide variants, are quite common, but they're not the only kind of event that can happen in the DNA. Another important concept is that of a frameshift mutation. And the way I think about this is, let's say you're a cell and you need to divide. That means you need to copy all 3 billion pieces of your DNA. And let's be honest, maybe we're going to skip 1 or 2 bases, right? I mean, 3 billion is a lot. So what were to happen if we were to take this little section of code, GAT, GAT, TAC, CTG? We can use our handy amino acid coding wheel here to say, oh, okay, well, this is consistent with the amino acid translation of DDYL. Cool. But if we were to lose that G, I mean, not just change it, but it's just gone, it's deleted, right? Well, what was, A was initially in the 2nd position, but now it's in the 1st position for the codon. The T, which was originally in the 3rd position, is now in the 2nd position, and the G, which is in the 1st position of the 2nd codon, is now in the 3rd. And we have completely changed not just the translation for this one codon, but all the codons that come afterwards. So what was DDYL is now MITC. Now, the good news here is that this will really only affect the gene itself, because basically the codons are reoriented at the sort of translational start site for every gene. It's really only the downstream elements of the gene which will be affected, but this can still be quite damaging. And we'll see a couple examples of this in Waldenstrom's. Another thing that's interesting to note is that these genes are really tools that have evolved over millennia. In fact, are highly conserved between different species. You can see here on the right, we have zebrafish, lamprey, chickens, elephants, dogs, mice, all actually having the same exact amino acid code, or at least very, very similar. Another thing that's sort of interesting, if you notice in our coding wheel, the 3rd base often has multiple options for the exact same amino acid. Well, this has led to some very interesting conservation in biology. So if we look at not just the conservation of the amino acids, but the conservation of the DNA itself, you can see that the conservation score for the first 2 bases in a codon is typically quite high, but the 3rd one is quite low. And this is because you can have a great deal of diversity sort of arise over time through genetic drift without having any amino acid consequences. And therefore it tends to be less conserved across species. And again, you can see this drop in conservation every third base pair. And this is really being driven just from the biology of how codons encode for amino acids. So that was a lot to take in. But I'm glad to tell you that we've now reached section 2, and it's really at the end of our basic science section. Now, this is a good point to pause and just talk about what exactly cancer is and how it happens. So we've talked about this idea of single nucleotide variants, frame shift mutations. They're occurring at a low rate all the time in your body. And for the most part, it doesn't do anything. There's no consequences to this at all. To the extent that there are, the cell is simply killed and replaced by a healthy cell. And it's that straightforward. But sometimes these happen and they can't be repaired. And sometimes the cell, in fact, isn't killed off and will continue to grow. And as these mutations accumulate over time, genes start to gain new functions and lose old ones. And this has consequences. And given enough of these events, the cells will eventually start to grow in an uncontrolled fashion. And when this happens, we call it cancer. So we can really think of cancer as being caused by a series of these genomic events accumulating over time, eventually winding up and causing something like Waldenstrom's macroglobulinemia. So what is the most famous and most frequent spelling mistake in Waldenstrom's? Well, it's actually a single point mutation. In this case, actually a T to a C or a leucine to a proline, exactly like the example we worked through earlier. This simple single base pair change completely changes the structure of a gene called MYD88. Now, MYD88 is actually supposed to be an adapter protein for a system called the toll-like receptor system. And you really don't need to remember this. What is important about the system is that it is a mechanism for detecting viruses and bacteria that are invading your body. It doesn't necessarily detect what kind of virus or what kind of bacteria, just lets the cell know that they're there and it's time to fight them. It's time to let the rest of your body know that they're there and recruit help. So we're thinking about growth, survival, and recruiting friends. That's really what this signal is going to give us. Now, typically, these receptors will activate and all cause mediate the dimers, or two different copies will come together and will signal downstream, ultimately activating a complex called NF-kappa B. This NF-kappa B complex will bind to the DNA and start a transcriptional process that actually promotes inflammation, doing all these things that we just talked about. Turning on new genes, turning off old genes, enacting this program that allows your body to basically respond to the presence of a bacteria or virus. Now, when it's mutated, this gene MYD88, it actually doesn't need the receptors to be activated in order for itself to activate and cause this constitutive signaling. And all your WM cells basically are feeling that they're constantly fighting off these bacteria and viruses. And that's really the signal that they're receiving pretty much 100% of the time. And this is true for about 95% of Waldenstrom's patients. Now, a lot of you are looking at this and being like, wait, you said this would be easy, and this does not look easy. This is a lot of words, a lot of arrows. What is going on? Do I need to remember this? Should I be taking notes? And I actually left this in here because actually one of the things that has happened is I've given this talk, or variations of this talk, actually not to patients, but to doctors. As doctors have come up and been like, yeah, I followed you up into this particular thing, and wow, that was way too complicated. So if you're feeling overwhelmed, you're in very good company. And so I'd like to actually take a minute and talk about what is it that biologists are trying to learn when they show you these kind of signaling diagrams, and really how you can think of them as patients with WM. Now, in reality, there's way more information and way more signaling details than anyone wants to think about in any of these cascades. The actual details of how things work are often very, very complicated. So it's really about what it is we want to communicate. And in this case, it's often as simple as, well, okay, there's a gene, it's called MYD88. It interacts with something called IRAK1 and 4, and another one called BTK, and it activates NF-kappa B. Done. That's all we want to say. Now, the downside of this right, is that we wouldn't be able to really understand why someone, for instance, might think that targeting TAC1 would be a really good idea in Waldenstrom's. You can see TAC1 here in the center diagram. Then all of a sudden, you know what? It actually makes sense. As a matter of fact, this TAC1 as a target has been investigated. It's a really good idea. It's not worked out great so far, but at least with this diagram, you can start to understand why people will be interested in this. And other times, of course, we need the whole enchilada. We need everything. But this quickly gets overwhelming, even for professional biologists. So we are always moving back and forth from simple to complex to very complex, back again, depending on what it is we're trying to communicate. But the most important thing to remember is that at the end of the day, a lot of these details really don't matter. We have MYD88, which is being activated, and it's starting this program called NFKB via Iraq and via BTK. And these results have actually been validated really across the world by many different groups using lots of different techniques. Again, really resulting in about 95% of patients having these mutations in MYD88. As a matter of fact, we can also detect this mutation in the majority of patients with IgM-MGUS. And this is a benign sort of precursor condition, sort of starts before WM. And this means that, there's additional events other than just MYD88 that's necessary to get full-blown Waldenstrom's macroglobulinemia. And this is a really important concept. We need multiple mutations in order to get cancer. And so later on in the presentation, we'll talk about what some of those are and how they work together to cause WM. Now, Guang Yang and her group made a really, really important discovery. And that is that while typically MYD88 is thought to signal through IRAK4 to NF-kappa B, it also signals through BTK, and the vast majority of signaling in WM is actually coming from this BTK-MYD88 interaction. Now, this is important because we have drugs that can target BTK and silence it. This includes the drug Ibrutinib, but there's also other ones, Dana-Farber and Acalabrutinib, and many others that are currently being investigated clinically. By silencing these, we can really start to block the vast majority of MYD88 signaling. I mentioned other mutations, and one of the other mutations that's very typical of Waldenstrom's are those mutations in CXCR4. They're in about 30 to 40% of patients, and unlike MYD88, where they're all pretty much the exact same thing, these are a whole group of mutations that accomplish something similar. They either are frameshift mutations, like what we talked about earlier, where everything downstream is scrambled, or there's something called nonsense mutations, which are special codons that actually cause the sort of signal the end of the gene. So by introducing this sort of translational stop sort of early in the gene, you can prevent anything further from happening. So truncating the end part of this, this receptor. So what is CXCR4 exactly? CXCR4 is something called a chemokine receptor, which is a fancy way of saying it's like a cellular nose. It allows cells to pick up a chemical gradient and follow it to its source. Now in this case, that source is typically the bone marrow. So CXCR4 can help cells move to wherever they're needed. In this case, again, typically the bone marrow environment, which should make a lot of sense in the context of WM. Now, what these mutations do is by taking off this sort of very tail of the gene, and you can actually see this protein, the protein structure here, it's really sort of sticking out away from the core part of the gene, away from the receptor part that binds on the outside of the cell, and also from the internal downstream signaling areas. And in fact, the gene can signal just fine with these mutations in place. What it can't do is be internalized. This tail sort of acts like a giant handle that allows the cell to bring it back inside the cell to turn it off. And without this handle here, it can't do that. And it will just sit on the cell surface and signal and signal and signal constitutively. Now this in turn has some consequences. There is a slightly different clinical profile. Patients with CXCR4 mutations typically have less lymph node involvement, but they also have some primary resistance to certain therapies. Now, the most famous example of this is the BTK inhibitor Ibrutinib, where their responses are typically not quite as deep, and are also greatly delayed. And this is one thing that we warn people about is if you have a CXCR4 mutation, Ibrutinib is actually a great drug. You still get a very nice partial response, just it's not as deep, and it can be severely delayed. It can be a month or more delayed, from the sort of typical patient who doesn't have a CXCR4 mutation, and just to know that going in. The other thing that people should be aware of is that those patients with CXCR4 mutations are often the first people to progress off. This is still after many years of therapy typically, so it's a good drug. It just is not as good for patients who have these CXCR4 mutations. Now, I've talked a lot about patients with MYD88 mutations, because they're 95% of WM, but there is a group, about 5%, who don't have them. Now, the first thing you should do as a patient, if you don't have a MYD88 mutation, is to make sure, in fact, that that is true. It turns out that not everybody has this T to C mutation resulting in a leucine to protein substitution. Now, the other mutations are only about 1% of Waldenstroms, but they do exist, and they also respond to Ibrutinib. So if you have been tested, make sure that that test really uncovered the entirety of the MYD88 gene, just in the outside chance. Again, it's a rare event, but once you're in this sort of MYD88 unmutated category, it really is, it's actually a fair amount more common because you're already in a group that's about 5% of the population. But there are some people who really don't have any form of MYD88 mutation at all. And luckily we've been able to study them. And one of the things we noticed is a number of the mutations that we did observe are actually mutations that are quite similar to other kinds of lymphomas. While this particular pattern is quite unique to WM, many of these mutations, again, are really being borrowed from, from other diseases. And it should come as no surprise that actually one of the real concerns for this population is that they have a much higher rate of transformation into aggressive lymphoma. And that's one of the things that doctors will be sort of monitoring you for if you are a MYD88 unmutated patient. The other thing is, when we take a bunch of these individual low frequency mutations together, we can start to realize that they actually all line up on a similar pathway, also one that's regulating NF-kappa B. However, these mutations are beneath the molecule BTK in the sort of signaling chain. This would indicate that we could actually inhibit BTK all day and it really wouldn't impact the cells. And this is in fact very consistent with what we see clinically. In the clinical trials using Ibrutinib, we saw very little to no response in those patients who were lacking MYD88 mutations. And these genomic findings really sort of help explain why that's true. It's also important to notice that this kind of observation can be very difficult to make. Here we already knew that the importance of NF-kappa B allowing us to see this, but it's not always so obvious how mutations work together along individual pathways and to sort of replicate signaling events, even though each one is different from each other. And this is one of the big efforts that we're putting in, trying to understand more of what these signaling events might be in WM, both for those who have MYD88 mutations and those who do not. We'll talk about that a little bit more later in the presentation. Now, the other big event I'd like to talk about today, and this is really the last one for those who are starting to feel a little overwhelmed, the sheer number of things we're talking about, but these are the somatic deletions in chromosome 6q. So, we talked a little bit about point mutations, like substitutions of individual bases, changing genes. We've talked about frame shifts that affect everything downstream in the gene, but sometimes actually just whole chunks of DNA go missing. And when that happens, we call them copy number alterations. Sometimes we get multiple copies like stacked up on each other end to end. Other times, again, just gone. Now, when this happens, obviously there can be consequences. Now we have 2 copies of every gene, again, 1 from each parent. So sometimes we can just use the other copy and it's no big deal, no consequence. Other times, having both copies is an important part of the regulation. And by losing a copy, we actually cut the gene expression in half. Now, one of the things we had noticed when we had done our whole genome sequencing studies is those patients who had CXCR4 mutations didn't seem to have as many deletion events on chromosome 6q. So Maria Luisa Guerra from our group, who is a wonderful visiting fellow from Italy, went back and worked with me to look at 5 genes that were in the 6q region and try to characterize them and see how they were actually playing out in Waldenstrom, see if we could get slightly better validation. So you can see the genes here, IBTK, FOXO3, BCLAF1, HIVP2, TNF-IP3, and she used individual probes to try to understand just the details. What percentage of cells had these? Were they all together lost, or were they just being lost sort of one at a time? And what she found is those people who sort of lost all 5 genes, in the majority of the cells, basically never had CXCR4 mutations, which was pretty interesting. They could be symptomatic, they could be asymptomatic, that didn't really seem to matter, but they typically didn't have CXCR4 mutations, and vice versa. Those who had CXCR4 mutations really didn't have these big clonal deletions in chromosome 6. Chromosome 6 deletions were also not observed in those patients who were lacking MYD88 mutations. Now, this is where it started to get interesting. Once we started to look and see how genes were being used in the context of CXCR4 mutations, and also how they were being used in the context of deletions in chromosome 6, we actually found that those gene sets overlap significantly. As a matter of fact, we're changing in about the same way and in the same direction. And this seemed to indicate that actually they're doing many, many different things, but some of their signaling in fact overlapped, even though the genes and the events are located in very different parts of the human genome. This sort of shared signaling event, you can think of as like having 2 different ways of going to the grocery store. You can take the highway, right? Maybe it's a little longer, but you know, it's the highway, so you get to go faster. Or you can go, you know, local roads, much slower, but it's much shorter. And there's different consequences for using each one, and you can choose. And you know, in the context of biology, in fact, both will often happen if there are 2 different options. And this is an example of that. It's called convergent evolution. 2 different pathways of getting to the same place. So this gene set is something that we're now very interested in trying to understand how it is it's contributing to the evolution of WM, because we now have 2 separate events that are occurring after MYD88 that are really contributing to this transformation from IgM MGUS into full-blown WM. So we've talked about these different events in Waldenstrom's. We talked about how sometimes they work together or don't work together. But I'd like to now move on to section 3 and talk about signaling networks and why they matter and what they are. Now, the most common form of signaling network in biology is a negative feedback loop. And it's a self-regulating biological network. Now, that sounds really intimidating, right? But actually, it's pretty straightforward. So it's the most similar, like mechanical analogy is a centrifugal governor. Now, okay, I'll be the first to admit that probably doesn't sound any easier, but actually the concept is very, very straightforward. It's from old steam engines. And basically what would happen is as the engine would spin, there are these large weights on lever arms. And so as the engine spun and spun and spun, it would be designed to also spin this core shaft, which had these weighted balls on it. And as they would spin, centripetal force would cause the balls to start to move out away from that shaft. And as they would rise, they would push the lever arm up as well. And that would actually then press down on, on the throttle valve, so restricting fuel, restricting steam, and that would actually reduce power to the engine, causing it to slow down. And that basically would mean that you could never get the engine going too fast. If it would start to go too fast, these balls would start to spin too quickly, it would start to rise up and push down that lever arm, sort of reducing the fuel intake, causing it to slow down again, and back and forth, right? Sort of keeping that engine always in that target zone. So this is a really important idea. And we have a very similar thing that can happen in biology. So let's just take 2 genes, gene A and gene B. We don't want to make this complicated, right? So what does gene A do? It makes a protein. That protein is called protein A. What does protein do? Well, in honor of a really good friend of mine from actually from grade school and high school, who was always telling me not to make things too complicated, not to overthink things, let's call them schmoopies. Every single time I was bringing sort of too much of my preconceived ideas to something, he'd be like, Zach, don't worry about it. It's just a schmoopie. It's a brilliant guy. So protein A makes schmoopies. Now, it turned out protein A also does something really important. Protein A is able to bind DNA right in front of gene B. and will cause gene B to be transcribed. Now, what does gene B do? Gene B makes protein B. And what does protein B do? It binds to the beginning of gene A and prevents it from being transcribed. Now, the way this works in biology is it's not like an on or off event. Actually, these proteins are constantly coming on and off the DNA. So the more protein there is, the more likely it is to be bound to DNA and either activating it or blocking it at any given time. So as the concentration goes up, in the case of gene B, there'll be more blocking of gene A. As the concentration of protein B starts to come back down again, there'll be that will sort of be released and gradually let up. Very, very similar again to this centrifugal governor that we talked about in the steam engine. Now, this again is the same concept. It's a self-regulating network. You have too much protein A, you'll start making protein B. Protein B will shut down the production of protein A. And this basically will make sure that you have a constant rate of SHMOOP production in your cells, assuming that this is something that's critically important. Now, why do we care about these networks? Why would we want to understand this? Well, understanding these networks allows us to understand how very, very different events link together to do the exact same thing. And this can be important in cancer because understanding the weak points of a cell means that we have to understand what it's doing and how these mutations that they've acquired are working together, and sometimes different mutations in different cells to accomplish the exact same thing. So we can imagine there's a mutation, say, in gene B that prevents protein B from interacting with gene A. This would cause uncontrolled SHMOOP production, right? Because protein A would just keep being transcribed with more and more and more SHMOOPs, but gene B would really no longer be able to regulate gene A. So that's one event. We can imagine a mutation in gene A that prevented it from binding to the promoter of gene B. Again, this event would cause gene B and protein B to effectively disappear. Again, with unregulated SHMOOP production. Finally, we could imagine epigenomic silencing. So we could actually just wrap up gene B so tightly that the cell just can't see it. Or we could put these methylation annotations on the gene, preventing gene A from interacting with it. This would also have the consequence of there being effectively no protein B to impede gene A's production. Again, leading to uncontrolled SHMOOP production. Did I do this whole slide so I could say uncontrolled SHMOOP production multiple times? It's entirely possible. But my real point here is that these are very, very different events that as we study them may only be in a few percentage of WM patients, but taken together are actually all doing the exact same thing and give us a clue of how the cancer is evolving. And in this case, that it's all, again, working together to create this sort of uncontrolled SHMOOP revolution. So what's an example of that signaling network in Waldenstrom's that actually has been worked out? So once again, I'd like to highlight my sort of brilliant friend and colleague, Guang Yang, who didn't just stop with the discovery of BTK and MYD88. He found that NF-kappa B was doing something else that was really important. If you remember the toll-like receptor system, that it's there to detect bacteria and viruses. Well, you know, that NF-kappa B signal, again, is not just an internal cellular signal, it's an external signal. They really want to let the cells know, hey, I saw this bacteria, I saw this virus, you need to help me fight this thing. You need to come here and help fight it. And this inflammatory process includes a number of molecules, but one of them is a molecule called interleukin-6 that is secreted by these cells. Now, interleukin-6 is interesting because there are interleukin receptors on the WM cells themselves. And this will lead to the activation of a gene called HCK. Now, HCK, which is activated when things like interleukin receptor picks up interleukin-6, it will signal downstream to AKT and ERK, which are really important sort of survival and growth factors for Waldenstrom's, like very, very critical. It can't survive without it. But it does one other really critical thing. HCK, it turns out, activates BTK, especially in the context of MYD88. That means that these MYD88 mutations drive BTK, drive IL-6, activate HCK, which makes more active BTK. This is called a feed-forward loop. It means every time we cycle through this, it gets a little bit stronger. It also means that maybe a neighboring cell, for instance, which doesn't necessarily have the same thing going on, will still be picking this up and get some HCK activation. As a matter of fact, we see this when cells develop resistance to ibrutinib. In fact, ibrutinib can no longer bind to BTK. These cells can still produce these IL-6 and may in fact help contribute to the resistance of cells who are actually still sensitive by providing this IL-6 and HCK activation to the whole microenvironment of the bone marrow. Now, these networks are clearly really important, and by understanding them, it allows us to know what to target. What should we be developing for new therapeutics in Waldenstrom's? Now, in order to help advance this goal, we've collected a series of 300 untreated Waldenstrom's patients and looked at actually the genetic sequence of all their genes. We've also collected from the same sample transcriptional profiles, so we can understand how these genes are being used. And then from a smaller subset, we also have different types of epigenomics, so methylation and also chromatin states. So looking at which parts are wound or unwound in individual Waldenstrom's cases, and also B cell and plasma cell healthy donors. Now, this is way more information than any one person could actually take on. And for this reason, we have assembled a team of pathologists, epigenomics experts, and also critically machine learning experts that can help us dig through this data to try to understand how it all connects. So we can see things like the convergent evolution we saw between CXCR4 mutations and 6q deletions, where they're actually accomplishing some of the same things through different mechanisms. Similarly, in the WM wild type group, where we saw multiple different types of mutations, all leading to the activation of the NFKB. We can use these machine learning techniques to try to both classify WM, come up with some predictions, but also study what networks are actually active in WM, how have they been altered from what we observe in healthy donor cells, and also how they might all fit together, sort of coalesce along individual pathways. And so this is actually a grant that we just got from the IWMF through the Roadmap grant system. And we're very excited about and look forward to sharing with you in the future. And this is, again, is all made possible by WM patients like yourselves who support the IWMF. So, you know, all this fancy machine learning is all something that's coming down the pike. But what, you know, we've managed to make a lot of really impressive discoveries actually even before this. And this is not just our work, this is people from all over the world looking at the genomics of Waldenstrom's, trying to understand how we can use what we understand about the networks to develop new WM therapies. And so this includes things like BTK, right? You know, Ibrutinib was only approved, you know, a number of years ago. It's not that long. And there's new BTK inhibitors, which are coming down the pike, and they're very exciting. We also have drugs targeting cell survival. This gets amped up in Waldenstrom's. Obviously, if the cell has a stronger survival signal, it's harder to kill. So there's a drug called venetoclax, which can help sort of counterbalance that. That would just finish a clinical trial in Waldenstrom's macroglobulinemia. We can also target the CXCR4 mutations by inhibiting the CXCR4 receptor itself. We just finished a trial with uliculpimab, and we're hoping to look at additional CXCR4 inhibitors and to see if we can effectively make these patients who have CXCR4 mutations behave as if they were lacking CXCR4 mutations and respond better to therapies like Ibrutinib. Finally, we didn't talk about the IRACs very much, but they are still signaling and they are independent of BTK. And so in our lab and through another IWMF grant, we are developing IRAC-based inhibitors that we can be used in combination with BTK inhibitors to help, again, really block out all the signaling that's coming off of MYD88 to see if we can actually make like better therapies that may actually get rid of the disease instead of just putting it in stasis. And finally, we're following up with Guang's HCK discovery to try to make HCK inhibitors and also inhibitors targeting MAP kinase and ERK to see whether or not they would be good for those patients who are you know, either resistant to Ibrutinib or to be used in combination with other drugs like Ibrutinib or venetoclax. So a lot of really exciting stuff that's been made possible because we understand these signaling networks. And again, this is all made possible thanks to the support that we've gotten from patients like yourselves. So I'd really like to thank all my friends and colleagues at the Bing Center for Waldenstrom's Macroglobulinemia, most importantly, my longtime friend and mentor, Steve Treon. Also, all the support we've gotten from the Bing Center, or the Bing Fund for WM, the International Waldenstrom's Macroglobulinemia Foundation, the Waldenstrom's Macroglobulinemia Foundation Canada, the Orzeg Family Fund, the Coyote Fund, the Bailey Fund, the D'Amato Family Fund, the Leukemia Lymphoma Society, an NIH Spore Career Development Award, an Ash Scholar Award, and of course, the Strategic Roadmap grants sponsored through the IWMF. And perhaps most of all, I'd really like to thank you both for your attention and for all of your support, which has made this work possible. Thank you very much, and thank you for joining me in this journey.